Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Hey everyone, Dr. D here, ready to go over the material that you need to focus on for the exam 1 for Biology 2420 at TCC. Now, here's the thing, I'm going to try to highlight some of the important things to know. However, please understand that I may miss some things. Um, by no means is this a comprehensive review. I don't have time to go over every concept in this video. This video is mainly there to highlight some more important things that you might want to definitely know, but by no means is it everything you need to know. And in fact, what I would do to prepare thoroughly is to go over all the handouts, go over those videos, those lecture videos that are on your Blackboard site, and study each one of those. Now, I do have something to share with you before we get started, and that's on the YouTube channel, Dr. D Explains Stuff. If you scroll down, I think I shared this with you in the orientation video, but just to remind you that there's a playlist under advice. You see this playlist, and the first video here is called How to Ace College. I would definitely recommend this video for studying for exams because, again, this video is me sharing my tips and tricks on how I succeeded in biology courses by studying the PowerPoint presentations, studying the outlines, and how to go about doing that in the most effective, best way. So I really, really recommend this video because, like I said, if you're ever wondering, well, how am I supposed to study for biology? How am I supposed to study for classes like this? Well, this is my best advice here. It's well worth the watch here. So let's go ahead and get started with our microbiology exam 1 review. So in this exam, we're going to be covering chapters 1, 3, 4, and 5. And let's go ahead and get started. So for chapter 1, one thing I want you guys to pay attention to is anything I've underlined. Okay, I'll try to point those out when we get to them. And again, I don't have time to go over every single little concept because that would be hours long review video, but I will flip through and at least highlight some things I'd definitely want to know if I was you. Okay, so let's get started. Chapter 1. I'm just gonna, like I said, I'm just gonna flip through till I find something kind of meaty, something juicy that we can digest, or I can tell you to study in more detail. Obviously, you should know the major groups of microorganisms. You should know, see how I've underlined this? Anything you see underlined, definitely know. You should know what viruses are and how they're classified. They are non-living. They are non-cellular. They do not belong to any of the domains of life. They are called small infectious agents. Let's see here. Again, I'm flipping through until I find something I find. Yes, obviously, this is an important slide here. The difference between eukaryotic and prokaryotic cells. There are eukaryotic microbes, and you should know what are the eukaryotic microbes. What are the creatures we study that are eukaryotes? And then, obviously, all prokaryotes are considered microbes. So those include the bacteria and the archaea, the domains of bacteria and archaea. And you should know that nucleus is only found in eukaryotes, right? Eukaryotic organisms. You should definitely know that eukaryotic organisms or cells, eukaryotic cells are larger than prokaryotic cells. Okay, eukaryotic cells larger than prokaryotic cells. And smaller still are the viruses. Viruses are the smallest. And they are not considered organisms. They are just infectious agents. What else do I want to know? Just flipping through till I find something meaty. through. Okay. All right, let's focus in on this slide. You should know that bacterial cells and archaeal cells are smaller than eukaryotic cells, as we mentioned just a minute ago. You should know that the domains archaea and bacteria are prokaryotic. You should know that they lack membrane-bound organelles. What does that mean? That means they lack any organelles which have a membrane, that are enclosed by a membrane, such as the nucleus. You're not going to find a nucleus in these prokaryotic cells, or mitochondria, or chloroplasts, or lysosomes, or endoplasmic reticulum, or Golgi. All of those things are completely absent in these prokaryotic cells. However, you are going to find ribosomes. You should know that you do find ribosomes and other organelles that are non-membrane bound. Okay. What else do we want to know? We want to know that the helminths, you should know helminths are worms. And we're going to cover some examples in a subsequent chapter. What else do we want to know? Oh, one thing you should know that you actually do need to know is anytime you see some scientist's name, someone's name, you should know what their contribution to microbiology is. So Aristotle, what was his contribution? Francisco Redi, what was his contribution? Louis Pasteur, what was his contribution? So again, I don't have time to go through each and every example and each and every concept, but if there's a name, you should know the contribution for the exam. Robert Hooke, Anthony van Leeuwenhoek. Okay, all of these are people to know. I'm just going to flip through now. All of these people, you should know their names. Semmelweis, he was a very interesting guy. Their name and their contribution. Okay. You should know the concept of aseptic technique, right? We're going to talk about that in chapter 3, I believe. Okay, Robert Koch. One thing you should know are, you should definitely know, or at least understand, his postulates. Remember Koch's postulates, the 4 postulates? And you need to know what those postulates are good for. What did those postulates show? And I underlined it here. Remember I said if you see it underlined in this video, you might want to really focus in on that for the exam. So those postulates allowed you to link a particular species of microorganism to a particular disease. Does that make sense? It allowed you to link the causative agent of a disease to the disease. So for example, through Koch's postulates, he was able to link the species Anthracis, Bacillus anthracis, which is a type of bacteria, to the disease anthrax. Okay, and these are the 4 postulates. This is the methodology that Koch developed to link a particular disease to a particular organism. Okay, and then you know about binomial nomenclature. You might want to know this. You see how I underlined levels of classification? Please know this for the exam. Do you remember the hierarchy of taxonomy? Domain, kingdom, phylum, class, order, family, genus, species. You should know this hierarchy in order from most broad to most specific, right? From most broad domain, there's only 3 domains of life. What are the 3 domains of life? Bacteria, archaea, eukarya. And then from those 3 domains, you have a bunch of different kingdoms. You have phyla, class, order, family, genus. And in case you're wondering a good mnemonic device so that you can memorize this list. One way is dude. King Philip came over for good spices, or at least when I was your age, that was the mnemonic device I used to remember this list. And just continuing on. And from there, I believe that's everything I wanted to touch base with you with Chapter 1. So hopefully that's all pretty straightforward. Nothing too challenging there. But again, just because I didn't specifically stress it in this video doesn't mean you shouldn't study. The best way to study, again, is to go over every slide, watch the videos that are on your Blackboard, and study. And if you want to watch that video I just shared at the beginning of this one, how to study in college, that's going to help you to really know what it means to internalize information and understand it. Okay, so let's move on to, why not save it? Let's move on to Chapter 3. Okay, here we go, Chapter 3. Let me get this ready for you guys. All right, Chapter 3. Let's go ahead and get started here. Chapter 3, this is regarding the tools in the laboratory, some of the laboratory techniques, media we use in the laboratory. All right, so obviously know these terms: inoculation, incubation, isolation, inspection, identification. You should be able to tell me, how would you go about culturing a bacterium? How would you go about growing it in medium? And how would you go about isolating a particular species of bacterium? Okay. And here is a summary of the general laboratory techniques. Don't read through this on your own. You should know what culture is, what medium is. Again, the term inoculation. What is sterile? You should know that sterile means free of any and all microbes or viable microbes. Incubation. Aseptic technique. You should know what aseptic technique is. It is a sterile technique, a sterile process by which you can subculture bacterium, move them around, handle bacterium, etc. It prevents what? It prevents contamination. Okay, you should know the types of media: physical state, chemical composition, functional type. Now you don't need to know, liquid, semi-solid, solid, in terms of physical state. With chemical composition, you should know the difference between synthetic and complex, which we'll touch on in a minute. And the functional type: general purpose, enriched, selective, differential. So let's get started. At least we'll touch on the important ones here. A liquid medium is basically like a broth, right? The bacterium grow inside of the liquid medium, and it is not great for isolating colonies, obviously. The bacterium just kind of float around in suspension. So a liquid medium is great for growing up a lot of a bacteria or a lot of bacteria, but it's not great for separating bacteria or really learning anything about the bacterium. Semi-solid medium is good for motility. You should know this for the exam, hint, that you can use a semi-solid medium for motility assays, or to determine if a bacterium is motile. That's a good one. What else would we like to know? Solid media, this is like your typical agar plate. You can spread bacterium on there. Synthetic medium, so know the definition of defined synthetic medium. Know the definition of complex medium. See what I underlined here? Contains at least one component that is not chemically definable. Okay, so definitely know that. General purpose medium, obviously know what that is, but look how I underlined enriched medium. Enriched medium contains complex organic substrates that fastidious bacterium require for growth. So you should know that fastidious bacterium require enriched medium to grow. What else do I want you to focus in on? Again, like I told you, I'm not going to go over the entire exam. I'm going to assume that you're studying each and every slide, but I'm going to go in and highlight the things that I definitely know that you should focus on. Okay. Discrete colonies, you should know that a streak plate method where you apply bacterium using a different zoned streak technique, which dilutes the bacterium down to individual pure colonies or isolated colonies, known as discrete colonies. You should know that this is a good technique for isolating bacterium from one another, from a mixture. If you have a mixture of bacteria and you want to isolate specific discrete colonies, pure culture colonies, the streak plate method is best. That's great to know. Again, see how it's underlined. Here, you should know the result of a pore plate. A pore plate is when you add bacterium to a liquid broth, but once you've poured that broth into a plate, the broth solidifies because of the agar inside of the broth. And the bacterium, because they were introduced into the liquid broth before pouring, those bacterium are now inside of the agar. So here, some colonies may grow deep. Here you go. Some colonies may grow deep in the medium rather than just on the surface. So actually a minority of the bacterium are going to be found on the surface. The rest are going to be inside of the medium because the medium was liquid when it was poured. And the bacterium were mixed inside of the liquid when poured. So that's good to know. What else should we know? Obviously, know the terms pure culture, mixed culture, contaminated culture. What could result in contamination? If you use poor aseptic technique, you can result in contamination. You can have contamination, which is unwanted microorganisms inside of your culture. What else? Flipping through till I find something. Okay, so here we're talking about microscopes and microscopy. Here you want to know what magnification means. Obviously, magnification is the number of times the number of times an image appears larger than it really is. Okay, so if a specimen appears twice as large as it actually is, that would be a 2 times magnification. What else? Resolution. You should know what the term resolution means. In fact, I think it's defined on a subsequent slide, but you should know resolution refers to the ability to discern between 2 very close dots, right? So if you have 2 dots, how close can those dots get before it appears as 1 dot? That's resolution. The better the resolution, the closer the 2 dots can get before it appears like 1 single dot. Contrast is how much the organism stands out against the background. All right. What else do we need to know? Resolution. Remember I told you resolution. You should know this term resolution. The capacity of an optical system to distinguish or separate 2 adjacent objects or dots, which is what I was talking about, or points from one another. You should know that the higher the wavelength, you should know this for the exam, okay? The higher the wavelength, like red light for instance, the higher the wavelength of light, the worse the resolution. The shorter the wavelength, the better the resolution you can get with a microscope. That's why, by the way, not only are, if you use red light, that's worse for resolution than if you use blue or purple light. But if you're talking about electron microscopes, remember that TEM or SEM microscopes, those have the best resolution, right? SEM and TEM microscopes have the best resolution, and that's because they use electrons instead of light. And electrons have a fraction of the wavelength of light. All right. What else do we want to know? Again, please study all this on your own, but I'm just searching for those things that I underlined that I really want you to know. Okay, I think that may be it. Oh, hold on. There are some things here. You should know the concept of a simple stain, differential stain, and specialized stain. Okay. Here, let's talk about special stains, for instance. A special stain. I want you to know that capsule stain and flagellar staining, these are examples of special stains. Okay. Whereas, what type of staining is a gram stain? That's a differential stain. And what if I just did a crystal violet stain and I stained all my cells with just crystal violet? What type of stain would that be? That's a simple stain. Okay, so just know the difference there. All right, that's it for chapter 3. Let's move on to the next chapter. All right, chapter 4. All right, let's go ahead and get started with chapter 4. This covers bacteria and archaea. I want you to know that bacteria and archaea have DNA, but their nuclear material or DNA is free in the cytoplasm. It is not inside of a nucleus. Only eukaryotes have the nucleus. So all of these have DNA. Bacteria has DNA, archaea has DNA, eukaryotes have DNA. However, eukaryotes are the only ones with the nucleus. That means eukaryotes have their DNA within a double membrane called the nucleus. What else? Bacteria and archaea, they have what's called circularized DNA, which means their DNA is actually in a closed loop, whereas eukaryotes have linear DNA. What else should we know? We should know that bacteria have a cell wall made of peptidoglycan. Archaea, although they also have a cell wall, it is not made of peptidoglycan. What else should we know? Eukaryotes. What about eukaryotes? Do they have a cell wall? Well, it depends. Some eukaryotes have cell walls, some don't. So you and I, our cells do not have a cell wall, whereas plants obviously have cell walls. Certain protists, like algae, have cell walls. Fungi have cell walls, right? So those are all examples of eukaryotes with a cell wall that you should know about. And again, bacteria and archaea have no membrane-bound organelles. So definitely know that peptidoglycan is unique to bacteria. That means only bacteria have peptidoglycan cell wall material. No other creatures besides bacteria are known to have peptidoglycan. What else do we need to know? You should know the general shapes of bacteria: coccus shape, bacillus, spirilla, spirochete, vibrio. You should know those shapes. And you should know that a lack of a particular shape means pleomorphic. Pleomorphism is the lack of a particular shape, and this is usually because the organism lacks a cell wall. You should know the terms staphylo and strepto. Staphylo means irregular clusters. Strepto means chains of. You should know the external appendages, the two major groups of these appendages, flagella and fimbriae or pili. You should know the parts of the flagella and their order. Okay, what do I mean? I mean, in what order do they present themselves from the cell? So first thing you have here is the basal body of the flagella. Then you have the hook, which connects the basal body to the filament. And lastly, you have the filament, right? So you should know those, and that's why I've underlined them here. You should know the flagellar arrangement of bacterium, difference between monotrichous, lopotrichous, amphitrichous, peritrichous. Know those terms. What else do we want to know? This term, although I didn't underline it, you should know the term chemotaxis. This is the movement of bacterium towards a chemical signal. Chemotaxis. Whereas phototaxis is the movement towards light. And obviously this happens in a series of runs and tumbles for prokaryotes. For flagellated prokaryotes, I should say. You've got run and tumble. Axial filament. This is another type of motility. Here's what you need to know. Prokaryotes have two main types of motility you need to know about: flagella and axial filament. This causes them to spiral. So if I were to ask you, you found, for example, a type of bacteria with motility, but this bacteria doesn't have a flagella, visible flagella, what would you say? Well, you could say, well, perhaps it has an axial filament. Perhaps its motility is corkscrew motility. Does that make sense? So just know that. That's why I've underlined this. Know that for the exam. It's good to understand these things. You should also know what the appendages are for. These appendages that prokaryotes have: attachment, mating, okay? And for motility, if it's a flagella, it's for motility. If it's a fimbriae, it's for attachment. If it's the sex pilus, it's for mating. So you should know the different functions of the appendages. What else should we know? What is a capsule good for? Some bacterium have capsules, and it's good for preventing against phagocytic cells, such as your white blood cells. Phagocytic cells come along, the capsule prevents phagocytosis. What else should we know? Definitely, this is a big deal in microbiology, is the difference between gram-positive and gram-negative cells. So gram-positive cells have a thick cell wall with thick layer of peptidoglycan. Gram-negative cell membranes have thin cell wall, thin peptidoglycan. And you need to know that gram-negative cells also have an outer membrane as well. So not just one membrane, but two membranes, an inner membrane and an outer membrane. And you should know that gram-positives, not only do they have, this is important for the exam, not only do gram-positives have a thick layer of peptidoglycan, but they also have these teichoic and lipoteichoic acids as well. Whereas gram-negatives, they have two membranes, the outermost layer of which being LPS, or lipopolysaccharide, and you also have these porins as well. So definitely know that. With the thin layer, obviously, the thin layer of peptidoglycan for gram-negatives as well. Mycolic acids are found in acid-fast bacteria. Again, I'm skipping through until I find something that I wanted to touch on. You should know this for the exam. You should know that all of these organisms from the domains of life, for the most part, they have ribosomes. And you should know that bacterium have ribosomes called the 70S ribosomes, whereas eukaryotes have ribosomes called the 80S ribosomes. So their ribosomes are different in structure. And you need to know that the 70S ribosome is comprised of two subunits called the 50S and the 30S. And yes, I realize that 50 plus 30 does not equal 70, but in this case, it does. The ribosome, the 50S large subunit, binds to the 30S small subunit to form the 70S ribosome. Okay, that's in bacteria. Just note that for sure. What else do we want to know? Endospores, know what an endospore is. Certain bacterium can form endospores, and this is for protection of in bad conditions. What else do we want to know? Now we're talking about archaea. Okay, that's it for chapter 4 that I wanted to touch on. Obviously, again, please study everything, but that's what I wanted to touch on. And now let's do the final chapter, chapter 5. All right, here it is, chapter 5, the final chapter for the exam. All right, let's take a look. Chapter 5, we're really talking about eukaryotic cells here, in eukaryotic microorganisms specifically. You should know that when we're talking about eukaryotic microorganisms, we're talking about the unicellular protozoa, the sometimes unicellular fungi and algae, and the always multicellular helminths. You should know that these are examples of eukaryotes. Definitely know that these are the eukaryotes that we're discussing. And you should know some of the structures of these cells. They do have proper membranes. They have proper membrane-bound organelles, namely the main one being the nucleus. You should know that animal cells don't have a cell wall. Plant cells do have a cell wall of cellulose. And for locomotion, you have flagella and cilia. Flagella and cilia. However, the flagella is not a filament like it is in prokaryotes. Remember, in prokaryotes, you had a flagella, but that flagella was a filament that spins. Remember, the basal body spins, and then you have counterclockwise rotation, you have clockwise rotation. You don't have any of that in a eukaryote. The flagella work differently. The flagella beat back and forth, and the cilia do a power stroke. Okay, so that's a big, big difference. And also the structure. You need to know that in eukaryotes, there is actually a 9+2 arrangement of microtubules that make up the flagella. So take a look at this. These green doublets are called, these are the microtubule doublets. And then there are 2 singlets inside of the flagella cross section. So you have 9 doublets surrounding 2 singlets. This is what they mean by the 9+2 arrangement of microtubules inside of the flagella of these microorganisms. Okay, so do you have the 9+2 arrangement in the flagella of a prokaryote? No. There it's just simple filament. Okay, cilia. Cilia are much shorter and they beat with power strokes. Okay, and they're found in single cell protozoa. Okay, they're found in protozoa. Prokaryotes do not have cilia. Cell walls. You should know which eukaryotes have cell walls. Well, you find fungi. Fungi have cell walls of chitin. Fungi, actually, I don't know too many fungi with cellulose in the cell wall, but chitin in fungi. Algae have cell walls. Plants have cell walls. Okay, so there are creatures that are eukaryotes that have cell walls. Okay, fungi, algae, plants, etc. Diatoms, remember diatoms? Those are a type of protist as well, and those have silica as their cell walls. All right. Let's see what else we need to focus in on. What is the job of the Golgi apparatus? You should know it's the site of protein modification and transport. Lysosomes. You should know that the lysosomes exist in eukaryotic cells. They float around in the cytoplasm, and you should know that they basically include these enzymes. They contain enzymes for digestion. Okay, they're basically a membrane enclosing digestive hydrolytic enzymes. Mitochondria. You need to know that the mitochondria is the site of what? Remember cellular respiration from BIOL1, cellular respiration? It's the site of cellular respiration, which provides the bulk of energy in the form of ATP to the cell. And here's something else you definitely need to know. The eukaryotes that have mitochondria, well, mitochondria contain their own DNA. It's called mitochondrial DNA. You need to know this. Mitochondria possess their own DNA. And actually, so do chloroplasts. Chloroplasts contain their own DNA as well. And this DNA is circularized, just like that of prokaryotes. Oh, and by the way, those mitochondria also have 70S ribosomes, much like prokaryotes. This is why we believe that mitochondria and chloroplasts originally, way back in time, were prokaryotes. Originally. Notice that the chloroplast also has DNA. You see this here. Chloroplasts also have their own DNA. Do all eukaryotes possess chloroplasts? No. You should know that the plants possess chloroplasts and the algae possess chloroplasts as well. And do you guys remember that in eukaryotes, the ribosomes are called the 80S ribosomes, and those are comprised of a 60S ribosome and a 40S ribosomal subunit, I should say. The large 60S subunit and the smaller 40S subunit, they come together to form the 80S ribosomal subunit. When we're talking about fungi, you should know that anything that starts with the prefix myco, anything with the prefix myco, like mycology, mycoses, this has to do with fungi. So mycology is the study of fungi, mycoses is an infection caused by fungi, etc. You should know the term hyphae. Hyphae is a long thread-like cell. That's distinct to mold, right? You've got those fuzzy mold. Those filaments are called hyphae. And obviously, fungi include the yeasts and the molds. The molds grow as hyphae. The yeasts grow as kind of roundish oval-shaped structures. And you should know fungal nutrition. How do fungi acquire their nutrition? They're saprophytes or saprobes, which means they obtain their nutrition from dead matter, dead organic matter, dead plants, dead animals. Now, fungi have spores, but are these called endospores? No, you should know that the spores of fungi are not a protective structure, they are a reproductive body. So the fungal spores are there to disseminate and to reproduce, not there for the same reason as endospores in bacterium. They're not there as protection. That's what is highlighted here again. All right, now there are two forms of spores, asexual spore formation you should know about, sporangiospores and conidiospores. Sporangiospores, here you've got the spores inside of a sac. Sporangiospores. You see the spores are inside of a sac. You should know that for the exam. Whereas conidiospores, the spores are outside of any kind of sac. What else do we want to know? Protists. What are the protists? These are your single cell, usually single cell or colonial organisms that are eukaryotic. Okay, they include, you can include the algae, the protozoa within the protists, algae and protozoa. Algae being the photosynthetic protists. Okay, so again, you should know that algae are photosynthetic. That means they contain the chloroplasts. By the way, do bacteria contain chloroplasts? No. However, can some bacteria be photosynthetic? Yes. There are a whole class of bacteria called the cyanobacteria, and those are photosynthetic bacteria. However, they do not possess chloroplasts because chloroplasts are a membrane-bound organelle. However, they do have chlorophyll, so they can conduct photosynthesis. What else do we need to know? Oh, you should know that protozoa have, these are, again, type of eukaryotic protists. You should know that they have various ways that they can move around. They've got pseudopods or false feet, like the amoeba. They can have flagella, the 9+2 arrangement flagella. They can have cilia. Okay. They can also be intracellular parasites. It's kind of interesting to know. What else should we know? These are the major things that I've found. But again, please make sure. Oh, worms. You should know the types of worms. Just be aware of which types, roundworm, pinworm, which types there are. Other than that, those were the major things I found that I really want you guys to focus in on. But to best study for the exam, again, I would recommend going through each slide, watching the videos that are on Blackboard as well, making sure that you review each concept and, again, look to my video on how to study for suggestions on how best to study. You can do it. These concepts are straightforward once you've gone over them a few times and you've really digested it, molded around in your head. You can do it. Hopefully this helped you to some extent. Let me know if it did. I can make another video for exam 2. But do your best, study really hard, and there's no reason you can't do this. I wish we could have face-to-face class, but in the meantime, this should suffice. And yeah, good luck. Do your best, and let me know if you have any questions. I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. 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 Dr. D,